class with a sentence that ends with a preposition.
Where do cells come from?
I could rephrase that for you. From where do new cells come? So I don't end it with a preposition, but I don't know that any of you actually care all that much.
That sounds weirder. Where do cells come?
From where do cells come?
Oh, you just said from where.
Yep.
Fun is not a word.
It is in my vocab.
So, good, excellent. Glad we had this detour in grammar. So, the question yesterday I asked you as your bell work was, where do new cells come from? And the same thing I'm asking you this morning. And you guys told me part of cell theory was the discussion I overheard as you were answering it. New cells come from pre-existing cells. And as it turns out, cells have a life cycle much like one we understand for humans or any other living thing, because cells, of course, because you know it from cell theory, are the smallest unit of structure and function that retains life. It's the smallest thing alive that's called alive still. If I were to ask you the life cycle of a human, what would you tell me?
80.
Not the average life expectancy, but just the life cycle. What does a human do over time?
Birth, baby, I think adolescence, I thought that everyone was teenager. Then you get old, you get older, you become easier, you lose all your hair, and you kind of that may have been.
So we appreciate that a life cycle looks something like, conception, growth in utero, where it looks like an alien kidney bean, then it gets born, and then from getting born, it turns into a child, then a bigger child, then, it got swaddled with a lot. Then it gets slightly bigger as an adult, and then it stays slightly bigger as an adult, and then they get smaller as an old person, right?
Yeah, and I'm understanding why. Why isn't that old people
Because
we could all be Michael Jordans.
there's a lot of different reasons that as you senesce, that's what we call that process, senescing, getting old. So a lot of stuff changes through here, right? And then, wow, spelling apparently today is not my forte. Then we have very little change, right? Like once you hit probably 25 to 45, it just kind of, like it's small changes, right?
Like you might have like a kid or
And then you get older and there's lots of change again. You senesce. You get older, things start to break down. You get smaller because you lose a lot of muscle mass as you get older. You lose cartilage as you get older. So you kind of, you age. That happens. We understand this as a natural process, but not necessarily for humans a cycle, right? Because this human here at the end isn't going to restart the cycle. It's more of like the process of life. Cells are going to do something very similar. There's going to be a period of a lot of change in a cell where a new cell comes to be. Once it reaches adulthood, it's just going to do what it needs to do to be an adult cell. It's going to fulfill its purpose in life. Whatever that purpose may be, it depends on the kind of cell it is. If it was a skin cell, it's going to perform the functions of a skin cell, and if it was a liver cell, it's going to perform the functions of a liver. So what it does here is entirely dependent upon what kind of cell it is. Now, some cells reach the point where they senesce, they get old, and they die. It happens. Some cells, that takes a really, really long time to get to this old person stage. Things that takes a really long time, things like your liver. Most of the cells in your liver are the cells that you got when you were born, like when you were conceived and you were developing in utero, and your liver develops somewhere around 18 weeks of development. So at 18 weeks, your liver started to develop. It continued to grow with you. It's still growing with your body size, and it'll max out when you hit like 18. Those liver cells are going to be the same liver cells, for the most part, that you have from now until you get real old up here. Your liver stops growing. You don't want it to keep growing. Once you reach full size, you don't want anything that's an internal organ to continue to grow because it doesn't have any place to grow. That's bad.
If that were to happen, could you explode?
You would rupture, yes. You're not going to like explode.
Rupture like a slow explosion?
Think of something more like a hernia. So hernias happen when all of the muscles that hold you together, because your outermost layers here are all muscles, right? To get to your internal organs, you got to go through your abdominal muscles and all that. Hernias happen when part of your intestines poke through your muscles. And they have to go in and stick them back in and stitch them up. That happens, that's a rupture. Very painful, very slow. You look like you're not real keen on the idea of a hernia.
No.
Same idea, your brain, think about it. Your brain, when you, again, it's one of those things, those cells developed, your brain cells, the stuff that makes up your brain and spinal column, that was really only like week 2 or 3 of development when those things started. When you were a wee little, well, okay, you wouldn't even have been this big. A baby this big is like,
You would have been a fetus.
this size is probably 18 weeks. So when your brain developed, you were smaller than, I can't even show you how small you were because it's so small. That's when your brain developed. Those cells gave rise to the nervous system cells. Those are the same cells that occupy your brain right now. Once you hit 25, your brain stops growing because your skull stops growing.
What if you...
Right? So those cells, they're going to last your lifetime.
So like skin cells, I don't like fleas.
Yes, those are the ones that are going to be fast. Your skin cells, you leave a pile of skin cells every time you leave this classroom. You don't worry too much about it, right? It's not something that's a problem. And your skin cells do this whole thing really quickly. So your brain cells, this takes like 85 years, right? Your liver, 85 years. Your skin, 3 days. Like, it goes that fast.
Is it like one stomach?
Yes. So it's going to speed up or slow down, and it speeds up and slows down with age too. Like when you're young in your skin cells, this is very fast, and when you're old in your skin cells, this is very slow. That's why old people start to look a little wrinkly, and their skin's not quite as young and fresh, and
Is that why when old people get cut, like sometimes they don't stop bleeding?
It's why they don't heal as quickly.
I can see how you rupture from like your intestines when your internal organs born, but the brain, like, it's not physically strong. What happens if that comes
You would die.
Would it just mash itself up within the skull?
No. So here's what happens. Your brain is floating around in a bunch of liquid right now. Like that's what prevents it from hitting the sides of your skull. You don't want it to hit the sides. Hitting the sides is called a concussion. If your brain swells for any purpose at all, it applies too much pressure to the brain tissue itself. That much pressure puts you in a coma. You lose consciousness. And unless they put in what they call a shunt is like a small little drainage tube in the back of the skull they put in to relieve the pressure. It drains out some cerebrospinal fluid. Unless they shunt it, you're going to die. So, yeah, brain, very bad. Like, your liver can swell a little bit. You're like, meh, no big deal, right? You just get like a puffy abdomen. Your brain's swelling, much bigger deal. We don't trifle with the brain. That's why brain tumors, very, very, very bad. Because that is tissue growing within the brain that shouldn't be growing there, that increases cerebrospinal fluid, which is why a lot of the first signs of a brain tumor are headaches. Because it increases, you can't feel your brain tissue. Like, your brain doesn't have any sensory, like, if I poked you in the brain, you wouldn't know. It's the pressure that you would feel, and that causes the headaches. And it's the increased pressure because there's increased brain material, because there's less space, same amount of cerebrospinal fluid. It's all very bad.
So kind of like a lot of animals competing for a niche.
There's a limited amount of space. Most definitely a limited amount of space. Cells are going to do something pretty darn similar to that. A cell is going to start out at the beginning of its cycle, and it's going to go through a series of growth stages. And it's in these growth stages also just going to live its life, like a human. Think about our human on the first slide. Yes, during childhood, the child is growing, right? But the child is also living its life. So here the cell is growing and living its life. It's preparing some essential materials. Consider this something like adolescence. Puberty hits here. So it starts to prepare for division. You're enjoying puberty that much? Here is something like in your 20s. So you're preparing for replication, you're preparing for marriage, for having a family, and then you enter that age or phase of life where you divide, where more come about. It's the same cycle, except in a cell, again, speed depends on the kind of tissue you are and the kind of cell you are. This particular process, the one that you're looking at, is specific to eukaryotic cells. So this only happens in our friends the eukaryotes. I am going to redraw this image in a way that I think is a little bit easier to understand. Hopefully my mad drawing skills are able to illustrate this is in fact a cycle. So this is the same thing, but instead of occurring on a straight line, I just took that line and bent the arrow to make a circle. It's going to start, if we start here, right there is where a new cell comes to be. Once it comes to be, the first thing it enters is a phase we call G1 or gap 1. Gap 1 is where the first initial stages of growth occur. You have a brand new cell. It needs to grow up a little bit. It's the childhood of the cell. Sorry, differentiation didn't fit. So gap 1 is where growth and differentiation happens. Differentiation is when a cell gets its job. So that means that the cell is a liver cell, and its job is to go about being liver. Or it's a neuron, and its job is to go out about being a neuron. So it gets its assignment here. Now, like I said, there are certain cells that do not need to divide. Your kidneys, your livers, your brains, anything that reaches maximum size and does not need to divide is going to actually kind of veer off of this cycle. Because if they pass gap 1, they're entering guaranteed cell division. We don't want them to guaranteed divide. That's not good for those cells. So they just take a little exit ramp off the side that we call G0, gap 0. That's where cells that do not need to divide go about all of their life cycles. It's where they perform their functions. It's the kidneys are going to be there cleaning out your blood, producing urine. Your liver is going to be removing any toxins. Your brain is going to be doing its thinking. They can re-enter the cell cycle if that tissue, so they can do this, they can re-merge back in. So it's more of a rest stop, really, like off the side of the freeway. They can merge back into the cell cycle if they need to divide. They would need to divide because that tissue sustained some kind of injury. So say you're in a car accident and your seatbelt holds you back from going farther forward, but in the process it bruises your liver. Right? It's holding you back so you don't fly forward, but that energy has to get absorbed somewhere. It gets absorbed by your body, bruises your liver. Your liver has to repair itself. So that's when they're going to merge back on, divide to fix the damage, and then once the damage has been fixed, they can merge back off and they no longer have to divide anymore. The next part of the cell cycle is over here. It's called S.
Wait, that means, like gap 0, they don't grow either?
They've reached full size, so they don't grow. S is synthesis. That's where DNA gets replicated. So DNA is replicated to prepare for cell division. Every cell has a fixed amount of DNA. So for example, in your cells, your human adult body cells, you have 46 chromosomes. Every cell must have 46 chromosomes in order for you to be healthy. That's how this works. So in order to make sure that each new cell that's made has 46, an exact copy of it must be made here. So we now have enough to divide between 2 cells. So DNA divides. During G2, which is just gap 2, during gap 2, we have the necessary growth and production of extra parts that are necessary for division. So any of the organelles that need to be replicated, anything at all that needs to be replicated for dividing out amongst 2 new cells, that happens here. The necessary hormones or chemicals, chemical signals, they all will replicate here. So that's gap 2. None of those things yet are considered cell division. Nothing has divided. All this is we add up G1, S, and G2. All together, these things are called interphase. Most cells are in interphase most of the time. So if you were to take a slide and look at cells, most of them are going to look like they're not doing anything because what they're doing is so tiny you can't see it because they're just living. They're not dividing. It's not until, it's not until we get to this section of the cell cycle that any division occurs. This section I've labeled M is called mitosis. Now, every year I encounter some of you who have in fact learned mitosis before. So I've got 1 nod, 2 nods. Any other of my friends familiar with mitosis? I got 3 shakes, 2 nods and 3 shakes. That's 5. Oh man, Mr. Murphy better watch out. I am so close to being a math teacher.
Let's do a project on that. One of my class failed it.
On math or mitosis?
We were like not familiar with it. She didn't teach us it, and then she just like gave us all posters. We went out and bought us all poster boards, and it was like an in-class project, and she forced us to finish it by the end of the hour.
I'm glad that you got you remember a great deal about it.
She was a terrible teacher.
Shh. I can't hear you.
This isn't 6th grade.
But it doesn't matter. I can't hear you. Yes. This wedge, much like interphase got divided into pieces, mitosis also gets divided into pieces. But to give you a general overview of what mitosis is, it's the process of the division of nuclear contents. Nuclear being the nucleus. So the division of nuclear contents. And it has a bunch of individual phases that will end with the word phase. Those of you who know them, prophase, metaphase, anaphase, telophase may sound familiar. Oh, project-based learning. Would you like to write a mitosis song?
Quick question. What's the difference between synthesis and GAP2?
So synthesis is when DNA replicates.
Okay.
So that's what's inside the nucleus, right? GAP2 is when all the other stuff is going to have to replicate. Because if you think about it, you're taking all of the organelles and dividing them amongst 2 new cells. So to make sure that there's enough for both cells, like mitochondria are going to need to divide and chloroplasts are going to need to divide, all that other stuff. And there's a couple of important chemical signals that have to get switched on. You'll notice that after mitosis, there's a truly tiny letter after M. I wrote like this teeny weeny itty little bitty wedge that says C. That's called cytokinesis. So that little C, where am I going to write it? Oh, down here. Cytokinesis is when everything but the nucleus divides. So mitosis, the nucleus divides. Cytokinesis is the actual division of everything else. And technically is not considered part of mitosis, but is considered part of the cell cycle. This whole thing adds up and is called the cell cycle. Mitosis has a lot of moving parts. So there's a lot that happens during mitosis. And it can be a little bit complicated. So before we look at the moving parts, we're going to take a look at a cell. Okay. A cell as drawn by Mrs. Bloomfield. It's round, so hi. I do.
To the room of sick AP kids that are trying to test. And Dean's in the hallway doing an interview, talking about hydrochloric acid and being a lab assistant for Mrs. Carl. It's just like, blah, blah, blah. I'm like, damn it. Go somewhere.
That's the moment you're like,
How's it going in here?
it's going well.
What are we learning about?
What are,
I was just going to say. It's like anaphase.
You don't know what to do.
well, they haven't learned their phases yet, but it is part of it.
Yes. I learned something when I sat in on the biology class. So. Yes. Thank you.
You're welcome. So I've drawn a few basic parts in here that you need to know. And I just want to make sure we remember them. This whole big thing is a cell. And this part is the
Cell membrane.
that says membrane. And this part is the and this is the these are usually in pairs at right angles. When we covered centrioles and we did organelles, I was like, oh, we'll revisit these later. They're a really important part of cell division. Well, here they are. And this is DNA that's been wrapped around some proteins, and we call it chromatin. And it's important that we know all these parts because every one of these parts has a very important role or job or thing to do in cell division. Centrioles, later in cell division, are going to produce these truly tiny fibers that we call spindle fibers. So centrioles produce spindle fibers. I like how when I run out of room to write things, I'm just like, oh, I'll just write the letters below it. It fits. Chromatin condenses into a structure that you guys may be familiar with. I don't want to flip because some of you are still drawing. So that chromatin is going to condense into a chromosome. So here's kind of what happens. So DNA is going to package itself up by wrapping itself up around these little proteins called histones. So we take DNA and we wrap it up around histones and nucleosomes. They look like little hockey pucks of protein. And those continue to wrap around each other and wrap around and wrap around and wrap around until they package up into a chromosome. And a chromosome is usually depicted as an X. And it's composed of two sister chromatids. Joined by a small structure called a centromere. So we have two sister chromatids that are joined by a centromere. You have 3 billion base pairs of DNA, which means 6 billion nucleotides in every single cell you have. 6 billion. When stretched out as just DNA, not wrapped around histones and nucleosomes, but as DNA itself, it's visible to the naked eye. You don't need a microscope. We can extract it from your cells, we can pull it out of your cells, put it on the end of a storing rod, and we can see it. So in order to fit that inside a cell, which we can't see, I know, now you're like,
How's that? That's not possible.
it is possible. It's all a game of packaging and folding.
What is this sister?
Chromatids. It's all about how you package and how you fold it. Yep.
What's that nucleocurd?
Oh, nucleosome. Yep. So it's all about how it gets wrapped up ever so neatly. When your body isn't using that DNA, it keeps it slightly packaged and condensed in the form of chromatin. When it comes to division, though, if we don't bundle the chromatin up into chromosomes, you can't guarantee equal division of all of the DNA. And remember, we said it's really important that every cell has exactly the same amount of DNA. So your body guarantees that this is going to happen by making sure that it packages the DNA as neatly as it can, as tidily as it can, in a very organized fashion. So organized, in fact, that technically this, let's call it chromosome number 1. You have how many chromosomes? You have 46 total, so 23 pairs. We always talk about them in pairs because you got one of the pair from mom and one of the pair from dad. This is one of your chromosomes. This would have all of the DNA that you got from mom. During S phase of interphase, an exact copy is made of it and stapled onto it so we know that those two things go together. They contain identical DNA. So sister chromatids are just a perfect photocopy of each other. And they're held together at the center so we know they go together.
What happens if they're not a
Then you have one cell that comes up with a mutation. So it ends up being essentially, well, some mutations are null and void, but some are bad. So this is one chromosome containing identical sister chromatids. That's an important concept to understand when we get to mitosis.
So if like someone that had Down syndrome with their chromosomes, isn't it because they have an extra chromosome? So would that have anything to do with that?
This process makes adult body cells. There is a very, very remarkably similar process that makes gametes, so sex cells. If something goes wrong with this in that process, that's how you get Down syndrome. So I should. So this process is intended to make somatic cells, so things that are not eggs, things that are not sperm. This is how we get new body cells that are identical to other body cells. Now you are absolutely correct. It is possible that mistakes get made in that copying process. Those kinds of mistakes, one of the ones that's easiest for us to understand is cancer. If we think about it, say, let's choose skin cancer because we just got back from spring break and some of us were sunning ourselves in sunny locations. If you think about it, your skin cells, we said they replicate very, very, very quickly, right? The faster you do something, the more likely it is you're going to make a mistake. Do you agree? Yeah. And how many times have you been told in grade school, haste makes waste? The nuns used to tell us that all the time. Really?
Sister Cecilia.
See? See? Did I not just say the nuns used to tell us that all the time?
I have not heard that before.
It astonishes me that you guys, I don't know, I feel like they used to tell us that all the time when we were little because they wanted us to like slow down and focus on what we were doing. So we did it well the first time. Same story here, haste makes waste. If you do it too quickly, you're bound to make mistakes. Well, your skin cells have to replicate very quickly so they can fulfill their jobs. Your body is excellent at catching mistakes during DNA synthesis, except sometimes they don't always catch those mistakes, and something gets changed, it gets mutated. So I say skin cancer because it's one that we understand. You guys know that you're supposed to wear sunscreen when you go out, right?
Well, I mean, if your body's not used to the sun.
No, you, regardless of your skin tone, regardless of how often you spend in the sun, you are always supposed to wear sunscreen.
Well, if it's negative like 3
What do you
degrees.
do in a pool?
Always supposed to wear sunscreen.
Really?
What if you're like, what if it's like a cold day outside?
What if-
Always
Where's
supposed to
sunscreen?
wear sunscreen.
What if the sun's not even out? What if it's nighttime? Do you
The
wear
moon's just a reflection
sunscreen?
of
Do
the
you
sun.
do you guys you understand UV radiation, right?
The moon's just a reflection of the sun.
UV radiation, anybody?
I heard of it.
So what happens?
In the winter time too.
When it rains, you put sunscreen
Serious, my
on.
mom does. She has cancer because she lived in Alabama.
See, I'm not making it up.
I can't hear her.
She is testifying the truth that it is in fact that you are supposed to wear sunscreen all year long, even in wintertime, because
She
of,
was really tan and she looked really pretty, so that's, no, it doesn't make up for it.
It doesn't make up for it.
so what happens is during synthesis, during that S phase of mitosis, or that S phase of the cell cycle, UV radiation can cause a mutation in DNA that causes this whole cycle to happen over and over and over and over again, even faster than it normally happens. So you get one cell that has one change called a thymine dimer. It's what happens when UV radiation replaces an adenine with a thymine in DNA replication. One single base pair change causes one single cell to no longer follow that circle. And instead, it does that circle on speed. Like it just fast forwards it 1,000 times. So instead of taking 2 or 3 days to replicate, that one cell is going to take a couple of hours to replicate. And that one cell becomes 2, and then those 2 become 4, and then those 4 become 8. And it continues to make identically mutated cells that become a tumor. Cell proliferation that's unchecked, right? That's what happens when this process goes wrong. That's why you're supposed to wear sunscreen even in the winter.
Do you wear sunscreen in the winter?
Yes.
All the time.
I have emphasized this in this room many times. Pasty. Paste.
So you just
Irish, Polish, German, paste. I have no choice. Besides, it's pretty easy. If you wear makeup that has sunscreen in it, and if you use moisturizer that has sunscreen in it, voila. You don't have to think about it. It's just there already. Yeah?
What are you using neither of those?
Sunscreen in the winter.
I just ran out of my last pack of makeup that you use.
Now, as we look at this process, you'll notice we have interphase here, and we have mitosis. And I just described to you what happens when something goes wrong. Well, your body naturally tries to defend against something going wrong. So every cell has checkpoints. It has a place that it stops, it surveys the scene, and it looks around to see or make sure that nothing has gone wrong. And most of the time, it catches it. So we end up with a checkpoint within G1, a checkpoint in S, a checkpoint in G2, and a checkpoint in metaphase. The DNA replication checkpoint, its job is to make sure that there's no damage in the existing DNA. So it takes a look at what's already there, and it's like, okay, everything that's here looks good. We're okay. In G2, it checks to make sure that the copy that gets made is accurate. So it's looking at it, and it's like, all right, you did a good job with this photocopy. You may proceed on. There's a checkpoint that occurs within mitosis that makes more sense once you know what goes on within mitosis. But this one basically makes sure that the chromosomes are going to be evenly divided amongst the cells. And our G1 checkpoint is just to make sure that growth has been permitted in that cell. It's checking to make sure the cell has permission to divide. Like, are you a neuron or are you epithelial. If you're epithelial, you have permission to divide. You may proceed forward. If you are a neuron, you do not. You must stay.
So why doesn't it see under?
Because here they folded it in with mitosis. A lot of times they will. The reason they do is because the last phase of mitosis and cytokinesis occur at exactly the same time. It doesn't have a checkpoint. If at any point in time something is wrong with these checkpoints, either the thing is fixed, the cell goes back and is like, oh, we can fix this, no problem. Or if it's an unfixable problem, it goes through a process called apoptosis. Apoptosis is scheduled cell death. What it means is the cell gets marked for death. It's marked, it's tagged. This cell will die, and it prompts the process by which that cell is dismantled for its parts. Every body is an amazing recycler. It has to be an amazing recycler. You get a limited number of resources, right? So that cell will be dismantled and used again, any pieces that are useful. Any pieces that are diseased, any pieces that are defective are disposed of. This is different than another kind of cell death. This is not what happens when you injure yourself. That's a completely different kind of cell death. That's the one that's caused by trauma. That's not a scheduled cell death. This is what your body does when it sees a cell and it's like, oh, that cell's a little bit too old to be useful anymore. Let's take it apart. We'll reuse it. So apoptosis occurs if any checkpoint comes back negative.
So is that what your body would naturally do? Like it starts to see those mutated cells?
Mm-hmm. Your body does naturally try and remove any mutations that it can. It's just not 100% efficient. It's remarkably efficient, but it's not perfect. Some mistakes happen. So apoptosis is scheduled cell death. I know it sounds terrible, like euthanasia, kind of is what it is. But